I'm sure you're wondering why my voice is so weird, but that's not important because today we are all talking about.
Hi, Professor D.
Well, hello, chap. Would you like to come into my lab?
Gee golly, sure thing, Professor D.
Well, hello there. Today I'm talking about DNA replication, transcription, and translation. But first, let me introduce myself. My name is Professor D. I think you know what the D stands for. But for those of you that do not know, the D stands for... I will now describe replication. Well, it all starts when DNA molecules unwind. Then the two sides split. Then the two nucleotides are added to each side until two identical sequences result. And now it's time for the D-dance!
Started from one cell and now we here. Started from one cell and now the whole dang family here. Started from one cell and
Now we explain transcription. During transcription in the nucleus, the information in a specific region of DNA is transcribed or copied into mRNA. It begins when RNA polymerase binds to the specific DNA sequence in the genetic gene that is called the promoter. The promoter site is the start location. RNA polymerase then unwinds and separates the two strands of the double helix to expose the DNA bases of each strand. RNA polymerase adds and links complementary RNA bases as it reads the gene. It eventually reaches the stop location in the DNA. The result is a single strand of mRNA. And now we will explain translation, which happens in the cytoplasm. Bury the body good. No mess-ups. Translation occurs in a sequence of steps. Involves three kinds of RNA and results in complete polypeptides or proteins. During translation, amino acids are assembled from information encoded in mRNA. As the mRNA codons move through the ribosomes, tRNAs add specific amino acids to the growing polypeptide chain. This process continues until a stop codon is reached and the newly made protein is released.
keep going.
Oh, dude.
That smells so bad. Stop.